Thank you very much. You've got a packed program, but I'm afraid you have to listen to some science before you get to that. But I do just want to spend a couple of slides introducing the Babraham Institute, which sits at the very heart of the ecosystem here at Babraham, and indeed founded the Babraham Research Campus 25 years ago. Babraham is best known for its three flagship research programs, signaling, immunology, and epigenetics, and I've put up there the things that those programs made an international splash with and that established their international credentials. Signaling, we discovered IP3 as a calcium mobilizing messenger, and we discovered most of the components of the PI3 kinase pathway. Immunology, we were in there at the start with LMB and humanizing monoclonal antibodies, and we've done a range of studies on signaling pathways and T and B cell development. Epigenetics, DNA methylation as the molecular basis for imprinting. That was done by Azim Sarani and Wolf Reich here at the Babraham Institute, and then ultimately has led to stem cell biology, but then also studies of the epigenetic clock and opportunities for rejuvenation. However, institutes don't stand still and they don't rest on their laurels, and we are constantly evolving. And that evolution, when the animation starts. There we go. Could be a long talk. That evolution reflects our evolving interests in aging and pathways which promote resilience as we get older. A healthy old age is the ability to adapt to a lifelong barrage of stresses. And so in signaling, we are increasingly interested in protein kinase signaling, ROS signaling, and also proteostasis. In immunology, we are now studying post-transcriptional gene regulation and how that regulates T and B cell development. But also, we're very interested in the decline in the immune function as we get older, using vaccine response as a model. And then in epigenetics, we are interested in how nutrients and aging regulate epigenetic states, how epigenetic states are established first during development, and how they erode across the life course, and how this undermines rejuvenation. And one theme which has emerged from all of that research in recent years is a cross-institute theme and focus around proteostasis mechanisms. Proteins control virtually every aspect of cellular life. The timely synthesis, trafficking, and degradation of proteins is critical for health of our cells, our tissues, our organs. And proteostasis is well known to decline with age and is both a biomarker of aging but also a driver of aging phenotypes. The accumulation of aggregated proteins is a normal part of aging. Obviously, it's exacerbated in cases of neurodegeneration. And so loss of proteostasis also drives diseases of old age where there are translational opportunities. Building on that, we actually established the UK Proteostasis Network, the website of which is up there if you're interested in it's very much a network for both academic and commercial bioscientists, and that was launched last year. And this really reflects our sort of national leadership in this space. And we've just actually held our second conference up in Dundee. So this is very much the direction of travel for the Institute, is studying proteostasis and various other resilience mechanisms which keep us healthy into old age. And I'd like to give you an example of one of those resilience mechanisms by going over a research project, which hopefully will soon be in press, which I think also makes a good example of the mechanistic approach to biology that the Institute has built its reputation on and continues to going forward. Now, this is a tale of two signaling pathways, so I need to introduce the first one. There we go. The integrated stress response. I noticed that there is a session on defense systems, the body's defense systems after this, and in fact, this is the cell's ancient defense system. The integrated stress response is a mechanism by which cells respond to a variety of different stresses by attenuating protein translation through the phosphorylation of EIF2 alpha, a translation initiation factor. This attenuation shuts down global translation, but it does allow the selective translation of certain key messenger RNAs, one of which is a transcription factor called ATF4, which drives a homeostatic gene expression program. On the right, I've put here one example. If you starve cells of amino acids, particularly essential amino acids, then they won't be able to make proteins. Therefore, they shut down protein synthesis because they don't want to make aberrant proteins. But they allow an upregulation of a homeostatic gene expression program through ATF4 to drive the synthesis of relevant enzymes to make those amino acids to get back to a homeostatic level. And this is an ancient pathway which is found in yeast all the way up to humans, and I think is critical for maintaining cellular and organismal health. Now, the second pathway I want to introduce you to may be more familiar to some people in the audience, which is the RAS-regulated RAF-MEK-ERK pathway. This has been a target for drug discovery for many, many years, and one of the great examples of a success in the clinic here has been the development of inhibitors which target the mutant form of BRAF, which is found mutated at high frequency in melanoma, colorectal cancer, and various lung cancers. And you can see both the success and the challenges of these drugs. The pictures I've shown you here, some of you may have not seen before. This is a man who was enrolled with end-stage metastatic melanoma. He'd failed on all therapies. He was enrolled in the phase 1 clinical trial of Babraham. There was a remarkable clinical response, but ultimately resistance emerged. That resistance can be mitigated or delayed through combination with MEK inhibitors, which target one step down from the pathway. There we go. And that actually allows a more prolonged clinical response. And in addition, these drugs are also now used in combination with immune therapies and actually is increasingly making melanoma close to being a manageable disease. One of the setbacks and one of the failures, however, of RAF inhibitors was that they failed to work in tumors with RAS mutations, which is 20% of all human cancers. So the reason they're successful in BRAF mutants is because BRAF signals as a monomer. However, in cases of wild-type RAF actually signals as a RAS-dependent dimer. So in tumor cells with a RAS mutation, RAS dimers activate RAF dimers to activate the ERK pathway. And quite bizarrely, in clinical trials of the BRAF inhibitors, although the melanomas with the BRAF mutation shrank, patients developed hyperplastic lesions, predominantly benign hyperplastic lesions, due to paradoxical activation of RAF dimers. And the way this actually works is that low doses of RAF inhibitor, the inhibitor binds to one partner in the dimer and inhibits it, but it causes an allosteric activation of the dimer partner. And it's only when you go to very high doses of RAF inhibitor that you actually shut down RAF signaling. And the consequence of this is shown here, which is an actual example of that, in which we've monitored ERK activation in a RAS mutant lung cancer cell line. And you can see if we use a RAF inhibitor, we observe this very nice bell-shaped dose-response curve for activation of the ERK pathway. And very unexpectedly for us, we actually saw an inhibition of cell proliferation. This is something that we did not expect at all, because in the clinic, you actually see hyperplastic responses, proliferation of otherwise normal cells. Now, I can tell you, through a lot of effort, we've actually found that 8 out of 10 RAF inhibitors that we've tested, including all of the clinically approved RAF inhibitors, do this. And we found that the inhibition of proliferation is not reversed by MEK and ERK inhibitors, so it's actually nothing to do with the ERK pathway. So was this a novel RAF effector pathway, or was it an off-target effect of RAF inhibitors? We felt it was important to understand this. So we did an RNA-seq experiment. We did an RNA-seq experiment in which we sought to identify gene sets which were induced by RAF inhibitor in a MEK-ERK independent fashion. And you can see the results of this here, and I'm going to cut to the chase, that the main signaling, the main gene sets that we observed, which were induced by RAF in an ERK independent fashion, were actually gene sets from the unfolded protein response. And I've given you 4 examples there. These are all classic UPR genes, ATF3, ATF4, which I've already mentioned, DDIT3 and DDIT4. Now, DDIT3 is also known as CHOP, which is a transcriptional target of ATF4. And these 2 transcription factors are critical in driving a homeostatic gene expression program, which decides whether a cell will adapt to stress and survive, or whether it will actually undergo apoptosis. We like a nice Western blot in the Cook lab, and here are several. This is just to show you that when we treat cells with dabrafenib, we activate the ERK pathway. That's the top data set. And this is prevented by a MEK inhibitor. Then if you look at the expression of ATF4 and CHOP, you can see that the RAF inhibitor, dabrafenib, is indeed inducing RAF expression, and that is ERK independent. It persists even in the presence of a MEK inhibitor. The other thing that this shows is that the other two pathways of the integrated stress response, IRE1 and ATF6, that's fine. The other pathways of the integrated stress response, IRE1 and ATF6, are largely not activated by RAF inhibitors. So this seemed to be very selective to the integrated stress response. And PERK, like GCN2, is another of these kinases which phosphorylates the translation initiation factor. So we used a PERK inhibitor from GlaxoSmithKline, a kinase inhibitor which inhibits the PERK enzyme, the PERK protein kinase. And we asked if this could reverse the inhibition of proliferation by RAF inhibitors. And the answer is it can't, even before it arrives. So the PERK inhibitor is perfectly able to reverse the inhibition of proliferation seen with ER stresses, such as tunicamycin and thapsigargin. But it has no effect on the ability of RAF inhibitors to inhibit cell proliferation. There we are. It was worth the wait. And so at this point, this was another unexpected result. We thought we'd got the story nailed down, and we thought that what we had discovered was that RAF inhibitors were activating PERK, but they weren't. So at this point, what do? We had to focus on other EIF2 alpha kinases which regulate this pathway, and I've already mentioned the fact that there are three others, GCN2, PKR, and HRI. Of course. Could be quite a long talk, actually. Well, anyway, what we discovered was that RAF inhibitors actually activate GCN2 in cells. And we were able to show using an ATP competitive inhibitor of GCN2, which is shown here, a drug called A92. This has no effect on ER stress responses, but it completely inhibits the it completely inhibits the expression of ATF4 by RAF inhibitors, and it also reverses the inhibition of proliferation by those RAF inhibitors. So is GCN2 actually required for these responses? We've used drugs, but drugs can have off-target effects, and so we also used CRISPR-Cas9 to knock out GCN2 cells. And I hope you can see here that the very strong increase in ATF4 by dabrafenib and also histidinol, which mimics histidine starvation, is completely abolished in the GCN2 knockouts, whereas the response to tunicamycin and ER stress persists. We focused then on a range of different activators of GCN2. We used 4 different RAF inhibitors. 3 out of those 4 activated GCN2. One of them, a compound called GDC0879, did not. But that activation of ATF4 expression here was, again, completely abolished by the GCN2 knockout. So this is a RAF inhibitor that are activating GCN2, and they are requiring GCN2 to activate the integrated stress response. We wanted to understand how those inhibitors were activating GCN2, and a very simple kinetic argument suggested that this was probably a direct effect. If you look at the very top blot on the left, you can see that dabrafenib causes a very rapid phosphorylation of GCN2, indicative of activation. And in fact, the time course of this is exactly the same as the activation of ERK by RAF inhibitors, which we know is a direct effect of RAF inhibitors binding to RAF. So we dared to think that perhaps RAF inhibitors are also actually GCN2 inhibitors, which paradoxically activate GCN2 dimers. And in fact, there was evidence in the literature, buried away in kinome studies in the supplementary data, that some early stage RAF inhibitors did have affinity for GCN2. We also notice in our cellular studies that if you look at the induction of ATF4, focused perhaps on dabrafenib as an example, you can see that at low doses of RAF inhibitor, ATF4 expression is induced very strongly, but if you go too high with those doses, it comes back down again. That's very much like the bell-shaped dose-response curves I showed you for activation of ERK. And we see this with several RAF inhibitors. And again, we think that this is evidence that RAF inhibitors bind directly to GCN2 dimers. They paradoxically activate them, and they only get inhibited at very high doses of RAF inhibitors when both dimer partners are occupied by the inhibitor. To actually get some evidence for this, we collaborated with Glenn Masson, who's a structural biologist who works on GCN2 and other EIF2 alpha kinases. And he used purified, recombinant, human crystallography grade, full-length GCN2 for these kinase assays. And I hope you can see if we add a RAF inhibitor into the test tube with this kinase, we see a dose response curve for a phosphorylation of GCN2 indicating it's been activated. And that goes up, it peaks at about 1 micromolar, and then it goes back down again. It's a bell-shaped response. GDC is one of the RAF inhibitors we find doesn't activate GCN2 in cells, and it fails to activate GCN2 in the test tube. Furthermore, we can repeat these experiments and include EIF2 alpha in the kinase assay as the substrate for GCN2. And you can see that 3 different RAF inhibitors, LY, encorafenib, and dabrafenib, drive the activation of GCN2 and the phosphorylation of its substrate, whereas the compound GDC fails to do that. Where do RAF inhibitors bind on the GCN2 structure? Well, actually, GCN2 has 3 potential ATP binding sites, and these inhibitors are all ATP analogs. The canonical kinase domain, but then a cryptic site in the pseudokinase domain and a cryptic site in the His-RS domain. So Glenn, being a structural biologist, used hydrogen deuterium exchange mass spectrometry to actually look at where these inhibitors were binding. And the solvent exchange diagrams for these experiments, which should be appearing any time now, actually showed that dabrafenib and encorafenib bind exactly in the ATP binding site of this kinase, exactly where you would expect an ATP competitive inhibitor to bind. So we can see changes in solvent exchange in the P loop, the activation loop, the DHG motif, and the autophosphorylation site. So these drugs are binding directly to GCN2. We actually used some in silico modeling to try and understand this process. And I will jump onto the next slide, actually. And what you can see on the top left is dabrafenib reaches into the active site of GCN2. It pushes tyrosine 651 up into the up position, which is critical for GCN2 activation. The LY compound does the same thing, but it does it by a slightly different method, whereas GDC bumps into an amino acid called the gatekeeper residue in the kinase domain, and it's blocked, so it can't actually do that, which explains why GDC won't activate this kinase. We use this structural information to mutate the gatekeeper residue, because this is ultimately the acid test of whether the inhibitor is binding right in the ATP binding site. And we changed the methionine to a very small alanine residue, or to an even larger, bulkier phenylalanine residue. And the first thing to notice was that the phenylalanine substitution is actually makes GCN2 a constitutively active kinase, whereas the much smaller alanine mutation actually opens up the ATP binding site, so it actually facilitates dabrafenib binding. And the consequence of this is that if you look at the Western blots at the top, wild-type GCN2 is activated by dabrafenib with a maximum effect at 300 nanomolar or 1 micromolar, whereas the 802 alanine mutation is now activated at 1 to 3 nanomolar, because there is a huge left shift there because we've opened up the active site and allowed the drug in. So this proves beyond all doubt that the RAF inhibitors at clinically relevant doses are indeed activating GCN2. So we set out to study paradoxical activation of RAF by RAF inhibitors. We've ended up over at the ribosome. Come on, you can do it. There we are. Where GCN2 resides, bound to the ribosome, monitoring amino acid levels. And when cells are starved of amino acids, uncharged tRNAs bind to GCN2 and activate it. But what we found is that RAF inhibitors can do exactly the same thing. Now, why should we care? Well, we've observed this response in all tumor cells that we've studied so far, including all of the tumor cells where RAF inhibitors are clinically approved. And this raises a very important question, which is GCN2 in the integrated stress response is an important pathway for allowing cells to adapt to stress. So is the activation of GCN2 by clinically approved RAF inhibitors a desirable property, or is it actually a bad thing? And that's something we're hoping to work on in the future. The other aspect of this, though, is also that turns out that GCN2 is required for longevity and stress resistance during caloric restriction. So reduction in calorie intake is the one gold standard method for increasing longevity and health span in a variety of model organisms. And it turns out that GCN2 is required for this in both C. elegans and in Drosophila. So we think there may be some relevance from this to having probes for investigating the role of GCN2 in healthy aging going forward. I'd just like to thank briefly the two people who really made the major contributions in my lab, Andrew Kidger, who's now a senior director at AstraZeneca, and Becky Gilley, who after 20 years is still not sick of working with me. So, and then very much the importance of collaborators at Liverpool, Dundee, Leeds, and Plexicon, the company who initially funded this project, who stuck with it through thick and thin, because they were the company who actually developed the very first BRAF inhibitor that went into the clinic and was clinically approved. And if you are interested in proteostasis, then you can find out more about the network that we've set up there. Thank you very much.